So, by my internet-enabled clock, we are at the 30 minutes past the hour. Going from west to east and in chronological order. Good morning to the people in the Americas. Good afternoon to the Europeans or the people in Europe amongst us. And finally, if there are some of you staying up late into the night in Asia and Australia and New Zealand, good evening to you as well. We will be giving this talk on Sophion's latest contractility platform, the Ethica M. We'll be starting in just a couple of minutes. We're just going to let the people to log on. But in this webinar, you'll be hearing from two experts in the field. So Dr. Ravi Vaidyanathan from FUJIFILM Cellular Dynamics will be giving you the first half of the presentation. And then the second half will be followed by my colleague, Dr. Lijo Cherian, here of Sophion, who's been one of the key members of the development team of this contractility platform, Ethica M. We do have some PDF handouts available. So on the right of your screen, you should see a chat box, a poll column, and handouts. So the PDFs of some documents relating to this Ethica M platform will be under the handouts. Through the webinar, as it progresses, there should be 3 polls intermittently coming online that if you do get a chance to spend, a minute or two just answering those poll questions as they land. And we do encourage you to put your questions into the chat box on that right-hand side of your screen. And we, with the 2 speakers today, will aim to try and get as many of those questions answered at the end of both talks. So we won't stop midway through and take a set of questions. We'll save all your questions for the end. If they are for a specific speaker, by all means, flag that up. And particularly if there are a specific question to 1 or 2 slides, maybe even mention the slides that you're referring to, if you so wish. But we'll try and address as many of those questions at the end. Please do that, put those questions into that chat box on the right-hand side of your screen. And we will answer as many of them as we can at the very end of today's webinar session. If you yourselves or, we encourage you to interact in the chat box, so don't feel it's just a black bin where you throw your questions and you'll never see the light of day again. By all means, we encourage you to interact in the chat box, and you may even have your own ideas or suggestions for potential solutions or answers to some of those questions. And we'll try and keep abreast of what's going on in the chat as we progress as well. We are now at the 33 minute past the hour mark. So I think without further ado, we should probably pass over to Dr. Ravi Vaidyanathan of FUJIFILM Cellular Dynamics, who can give you the first half of today's webinar. Over to you, Dr. Vaidyanathan.
Thank you, Damien. Hello, everyone. Today I'm here to talk about how you can advance cardiac discovery, drug discovery, using our cells, and then the second part of the talk will talk about how our cells are used on the Ethica M. So a little bit about FUJIFILM Cellular Dynamics. So we were the first iPSC company launched in 2009. We are based out of Madison. FUJIFILM acquired us in 2015. And they are, of course, headquartered in Tokyo, Japan. FCDI, that is how it goes after the acquisition, is primarily divided into 2 divisions. So we have the discovery research and the cellular therapeutics. Today, I'll be talking mostly about discovery research that we do here at FCDI. Our core competencies are listed here. I'll highlight a couple: iPSC reprogramming, genetic engineering, and then scalable manufacturing. Those are the key highlights that I would like to do. And then why use iPSCs? Because we feel it addresses numerous challenges that exist with existing models that are available, enabling NAMs, which is a very popular term nowadays. So looking at primary cells, transform cells, or animal models, they all have their drawbacks. Mainly with primary cells, you will see there is limited availability, there is variability in donor to donor and the quality of the cells. There is phenotypic instability and a limited characterization. With transform cells, they do not really recapitulate relevant tissue or cell biology. They lack key functions. And then animal models, they do not represent human biology. Working with them can be resource intensive. And of course, there are animal welfare issues. So with the use of iPSCs, we can overcome a lot of the limitations, mainly making ourselves, by addressing key functions such as availability, functionality, reproducibility, and translatability. We, as I said before, we are using our proprietary reprogramming techniques to engineer cells into iPSCs and then can differentiate in different cell types that can then be cryoed and then thaw and use in your workflow. We are platform agnostic. We have a list of instruments here that we have worked with in the past. I'll address this later in one of my slides, but over the years, we have launched over 250 products encompassing both healthy, normal, and disease cell lines. And today, my focus is going to be in the cardiovascular portfolio. Highlighted here are cardiomyocytes squared, the endothelial cells, and fibroblast. So as I said before, our company started in 2009. The first product, which is our flagship, it was launched. It's called iCell Cardiomyocytes. They are the first generation of cardiomyocytes. They are highly pure. They're primarily used in drug discovery and disease modeling. They're ready for assay 10 days post-thaw. Cardiomyocytes squared is the second generation. Similar to our first generation, it is highly pure. This line has been validated in multiple studies, such as the HESI and the JIXA, which is the Japanese SEPA study, and then also by the FDA. And there are many publications identifying that. The reason people love these cells, mainly for safety pharmacology and high-throughput screening, is because they're ready for assay 4 days post-thaw. So you can thaw them on a Monday and assay them on a Friday. No weekend work. And then these cells come with their own media, which is DMEM-based and serum-containing. They've been used in all of these studies that I just mentioned, and they're typically used to model how drug behavior in a physiological settings. We also have a serum-free media. I'll touch on that later on. Using these cells, most groups, at least in the past, I would say 10 years, have been doing monoculture for the most part and trying to do disease modeling using endothelin 1, that is what is shown on the top left. You can also use it for clinical investigation, where we're showing an example here that was published in 2016, where sofosbuvir and amiodarone in combination can cause arrhythmias, and that was identified in this publication. We also worked with other platforms where you can mature our cells using different substrates or using complex cultures. And then, as I said before, it can be compatible for high-throughput screening, and that is what is shown in the proarrhythmia assessment slide. This was done on a high-throughput system, basically demonstrating that our cells work across basic biology, can be used for drug discovery, disease modeling, safety pharmacology, and clinical investigations. But the heart is not just made up of cardiomyocytes. And this is what the slide talks to you. So this is a publication that came out in 2020, published in Nature. And on the left-hand side, you can see a heart where there are certain areas that are highlighted. So tissue biopsies were taken from these different areas and from different 14 patients, and they looked at the composition of the different cell types in the heart. Of course, ventricular cardiomyocytes are the majority, as you can see on the pie plot on the right. But the other cell types that are important is cardiac fibroblast and endothelial cells. And as I always say, every cardiomyocyte sees an endothelial cells because that's how they get their nutrients. So with this in mind, the field is currently moving to more complex models, and we actually have endothelial cells and fibroblasts all available in the same donor background as our cardiomyocytes. So here, just showing you basic morphology and purity and identity of our iCell endothelial cells. They are highly pure. They have the right morphology. They express the right endothelial markers and can be used in co-culture with our cardiomyocytes. We also launched our iCell cardiac fibroblast, and they also express and have the expressed key markers. I'll touch on that in a bit, but they have the right morphology. They lay down collagen, they express connexin 43, and they don't express preustin mainly because these are not activated. And you can also see that from the flow charts that are shown on the left bottom, where they're highly pure by T7 marker, and they are alpha smooth muscle actin negative, which is a marker for activated fibroblast. So with these three cell types that I just talked about, we also need them to work together in a medium that can support all three cell types. So for enabling that, we've launched our iCell cardiac co-culture supplements kit. So you can just combine the supplements with either our serum-containing cardiomyocyte maintenance medium or our serum-free medium and use them for your experiments. The main difference between the cardiomyocyte maintenance medium and the serum-free medium is the absence of serum. You can culture the cardiomyocytes in the serum-free medium for up to 7 days, which is usually what the window is. And most people who use the serum-free media currently are using it to understand how drug interaction in a serum-free composition. The iCell cardiac co-culture supplements kit can be used in collaboration with mixture with the cardiomyocyte medium or the serum-free medium, and culture the cells for up to 3 weeks in co-culture. We also recommend that if you're interested in fibrosis assay, you can use the co-culture supplement with our ICMM and activate the cardiac fibroblasts in your workflow. So the reason for showing you all these slides is that the field is now moving away from monoculture in 2D to complex cultures. And there have been a few models that have been shown before. So the most popular ones are listed here. So you have the BioWire platform, then you have spheres that I'll talk about later, and then you also have the ring technology. But on the right, I wanted to show you that in 2018, AstraZeneca published a paper where they used our cardiomyocytes in combination with primary fibroblast and endothelial cells and created these spheroids. And they were able to show that the spheroids express cardiac troponin I, and when treated with toxins such as doxorubicin and sunitinib, which affect structural, that can cause structural toxicity, they affected the expression of cardiac troponin I. And to that end, what we have done is we have come up with a protocol where you can combine the three cell types in a specified ratio, so 65% cardiomyocytes, 15% fibroblast, and 20% endothelial cells, right out of thaw, to form cardiospheres, and of course using the co-culture supplements. And you can assay them in a high throughput format. And that is what is shown here. So using the protocol just mentioned, combining the three cell types, you can make single nice cardiospheres in a 96 or a 384 well format. And we have tested multiple plate types to show that it works efficiently on different systems. When we also tested them with cardioactive compounds and compared them with DMSO, we saw the appropriate response in these spheres. So basically, E431, which is a hERG blocker, prolonged the calcium transient duration. BK, which is a calcium activator, also prolonged the calcium transient, but at a different phase. As you can see, the APD 30 and 50 are more pronounced. Isopaternal and digoxin are positive inotropes. And you can see that they sped the, so they had a chronotropic effect. And I'll address that in the next slide. They also had a inotropic effect on the inotropic response. And then verapamil, which is a blocker of calcium, blocked the calcium current, reducing the calcium transient amplitude. So the whole point being iCell cardiospheres respond to compounds known to modulate cardiac activity. So these spheres, if you culture them for up to 14 days and change the media every other day, and it can also follow a weekend-free workflow, you will see that when you challenge them to positive inotropes, here we are showing you 3 different drugs, but on the left-hand side, the black is the DMSO control, and with isopaternal, not only the calcium transients are faster, but also bigger, and that is what is shown on the right with multiple positive inotropes such as digoxin, dobutamine, epinephrine, and isopaternal. The other aspect, if you see the 2 colors, they are from 2 different donors. So we compared them with 1434 cm squared donor, or compared them with the regular cardiomyocytes, the first generation. So we see this across donors too. This is the last slide, and I wanted to spend some time mainly addressing how our cells are being used. So this is a very interesting study that came out early this year. It was a collaboration between Takeda and Wallow Health, where they were comparing how the gene expression of different genes are different between human heart tissue, 3D engineered heart tissue, and 2D humanized iPSC-derived cardiomyocytes. And what you will appreciate here is that if you see the 3D engineered heart tissue tends to have, in most instances, higher expression or similar expression to that of adult. So if you look from the left, if you look at calcium handling proteins, the 3D tissues tend to have similar or higher expression of the genes that are listed there. The T-tubule proteins are the same. Both ion channel expression is also higher. For example, if you see column 2 where there's KCNJ2, that is IK1. IK1 is really expressed highly in the 3D ECTs. So this was using our cells. So our cells are capable of getting really mature and being able to reproduce adult cardiac phenotypes, and that is what is shown in this slide. So the final point that I wanted to make is our cells are compatible with most systems in 2D and 3D. There is definitely value to working in 3D. And in combination of cardiomyocytes, fibroblasts, endothelial cells definitely help make the cells more mature and have the right response. So with that, I would like to pass on the slides to Damien back again.
That was perfect and really on time as well. So thanks for that too. Just a reminder to the people who may have come on late, we will be getting Dr. Vaidyanathan and my colleague who's just about to come on now, Lijo Cherian. We will be getting both back at the end to answer all of your questions. So do get those questions coming into the chat box on the right-hand side of your panel. I don't want to take too much more time. So I will pass over to my colleague, Dr. Lijo Cherian, who will take the second part of this webinar, and we'll be back at the end, hopefully to answer as many of your questions as we possibly can. Okay, bye.
Hello, everybody. Thanks for joining. From which part of you are the world of you are joining in? Good morning, good evening, good afternoon also. So I'm going to talk about one of our new platform we just recently working on also. And before going into just a back history about Sophion, which started 25 years before. So So back in 2000, we started by a group of electrophysiologists who has been very much interested to develop automation, bring in automation to patch clamp, which itself still is one of the challenging techniques to earn or get the expertise in. So we have been quite pioneered in developing automated patch clamp platforms and have been catering our customers, mainly big pharmas, large pharmas, but also the CROs, even the academic labs. And we do help them to build up their science and generate that data, which they could more believe in that data, much more faster data, and have confidence in their data. So we have been doing this for past 25 years. And since being a part of this drug discovery pipeline for a long now, we have been closely interacting with our customers who have been giving us a lot of feedbacks about the challenges they face in this field. And one of them, the key of them, is actually a lot of drugs fail in this whole pipeline because of its safety liability. One of them is the cardiac safety liability. And what are the reasons behind that is actually the poor predictability or the transferability of the results you acquire in your pre-drug discovery phases, which is usually done in 2D systems or is done in single cell systems, and then you sort of translate into humans or larger animal models. And there, the chances of failing your drugs are quite high. And given that the authorities have been also demanding for bringing in a shift, bringing in a change and reducing the animal works and replacing it with more humanized model system, as my previous speaker, Ravi, also mentioned about having these iPSC-derived cells. So this has been something of our interest as we come back to this whole discovery pipeline. Until now, we have been catering that early drug discovery phase using our automated patch clamping, helping you to develop your targets or even develop your screened compounds to your targets there. And then the transition to much more clinically relevant model system, that's where the gap was there. And there, the growing demand of organ-on-chip sort of platform is increasing. And this is what I would like to bring up today. And what we would like to introduce is the Ethica M platform, our solution for this organ-on-chip platform. So I will go details about these, but some of the key features includes high throughput, because this has been the value in this whole discovery pipeline, the throughput scale. Usually we say that the throughput is required for pharma. It's not the case. Even nowadays, academics also need throughput because they work with different groups and different assays also. So the throughput has been key here in our platform. You can have 9 different assays or 9 different plates running simultaneously on this platform. As I mentioned, this is more using humanized model systems, so stem cells and iPSC-derived cells, which comes at a cost. So we made sure that our data points are also cost effective by using least amount of the cell, but getting the much more better data out of it. And automation has been key because since when you work with this high throughput and big number of assays, you need also automation to make it more reproducible and robust assays. So this is how we came up with Ethica M. So Ethica M is not just one component. It's a total solution which Sophion provides you. The solution which starts from producing your engineered tissues right to your acquiring the data, analyzing and interpreting the data. So that's a whole solution that comes with. So some of the components used for Ethica M as a solution includes the pipetting robot, which helps you to make engineered tissues more reproducibly, much more robustly. The platform itself, which is used to measure and acquire data, which sits in a cell culture incubator, usually 125 liter incubator. At the same time, a software package which helps you to automate your data acquisition and also your data analysis. And the N-chip consumable, where all the magic happens. The engineered tissues are casted and generated, matured, and all the assays are done on your N-chip consumable plates there. And as it has been always in Sophion's DNA for past 25 years, we also support our customers once the instrument is there with developing your assays, making sure your assays keep running. At the same time, our service engineers also make sure the machines keeps running there. So going into details of each of these components quickly, about the optical measurement system. So this is an optic-based system. So it houses 4 cameras, which acquires the images and videos of the engineered tissues. And it's a wide field, so you don't need any dyes or anything. It acquires in a bright field area. The 4 camera, high-speed camera, so you get data points at a very good high sampling rates. And as I mentioned earlier, it can house 9 96 well plates. So it's a standard 96 well plate size, and you can have 9 different assays running parallelly. And since its name says Ethica M, and M stands for muscles, so we are working with contractile tissues. So electrical pacing is something key for the contractile tissue. So you have an integrated electrical pacing units. It's not like a lot of cables are hanging around. Everything is sort of integrated inside this platform. And as I mentioned, it sits inside a cell culture incubator, 240 liter cell culture incubator. Moving to the second components, that's the N-chip, which is a standard 96 well plates. So the outer frame dimension is a standard 96 well plate. So it helps you in future to move across your different platforms in the lab. For example, a pipetting robot, or it can be also used for some imaging platforms. The other key of this is these N-chip consumables are being produced here. And so it also has integrated electrodes, spacing electrodes. There is no need for putting in extra electrodes or anything usually comes up. Then you need to have the maintenance and all. You don't need to. It's all integrated inside these 96 well plate. And inside each of these wells, you have these 2 3D printed mini well structures. And inside these structures, you have these 2 pillar structures onto which the engineered tissue sits. And so basically, if you do the math, it gives you 192 tissues in one 96 well plate times 9. So that's around, if you run all the 9 plates, that's around close to 1,700 tissues running different assays simultaneously on this platform. Second thing I would like to highlight is also it's not a PDMS-based material, unlike some of the platforms which use PDMS. And we know from our customer base that they have been using PDMS, and we know that it has some of unwanted drug absorption issues. So right from the beginning, we stayed away from PDMS. So it's a non-PDMS-based material. That gives you also an option to customize, because we are working with contractile tissues. Some tissues are more contractile, some tissues are much more robust. So we can actually control the stiffness of these pillars, which gives you option to customize your plate to your assays or to your cell type. When you see these N-chips, that's a small size in 196 well plate, you have those 2 mini wells, and casting tissues manually into that itself is a challenge. So you want to make sure that you get robust tissues every time you generate an N-chip at your site. So what we use is a pipetting robot. So what the pipetting robot does is it premix. If you need to load your metrics and your cell of interest and load a certain protocol which is predefined. And the pipetting robot mixes these two and starts dispensing inside these mini wells precisely with micrometer precision. And that helps you to get you good tissues in your plates. And since this sits outside an incubator, so it has also an integrated HEPA filter and UV lab, so that gives you that aseptic condition to generate your plates. Not just to generate your plate, this platform, this robot will help you to exchange your medias regularly. At the same time, also make your compound plate for your endpoint pharmacological assay. So once you have your plate, you move into the platform which sits inside the incubator. Now starts the other job. That's to acquire your data. These sort of engineered tissues is not just one day assay. It's a day which is going from few days to a month also. So how to manage or how to acquire these data so regularly? So what we have done is our software actually, you can schedule the things upfront. You can schedule capturing of videos, images, or even paste the tissues. Say from day 5 to day 6, I need to paste these tissues in a certain pacing frequency. You can load it, run it, and just walk away. So the system will go regularly to take all those videos, do the pacings for you when you are back at home. So the scheduling helps you to automate your workflow for regular acquisition, data point acquisitions there. And then you can have also control, because you control access, because you have 9 sites, 9 different assays. Maybe it's 9 different groups in the same department using the platform. So you can isolate your data from other users and be specific to your purpose. So you have controlled access there. So it's going to collect a lot of data. So you also need an automation to analyze those data. So that's where the analyzer also kicks in. All your data is stored locally on your computer. Nothing is on cloud. So it is on computer. So as soon as the system acquires a video, the analyzer kicks in and starts automatically analyzing all your videos and generates your contractility data. So the next day morning when you come in, you have your plot. So what you do is you group your tissues of your interest, you select them, and you get the plot of your interest. So this is the quick overview about the whole solution. Again, as I said, we have the assay support and also the technical support to make sure your assays keep running and our machines also keep running. So I would like to get into now the more interesting part of also the science. So here I'm going to present about the data of generating engineered heart tissue using the iCell cardiac myocyte square from FUJIFILM. We generated these engineered heart tissue with iCell's cardiac myocyte square and also iPSC-derived cardiac fibroblast. So before getting into that, how the whole workflow starts. So we have an SOP developed in-house here where you can actually freshly thaw your cells. You don't need to sort of pre-culture them. You freshly thaw your cells, you dispense them into certain wells inside a PCR plate. We also have a gel matrix mix. You can also use your own gel matrix, and you dispense into these into predefined positions on your PCR plate. Load this PCR plate inside this Ethica M pipetting robot. Once it is there, you just need to start your protocol, and the pipetting robot starts dispensing the cell gel matrix inside these mini wells. Within a few minutes, the whole M chip is being seeded, and this is now you need to move it onto this optical measurement system, which is sitting inside the gantry. There you have already scheduled your actions, which includes taking videos or taking images, pacing your tissues for next few days. All you have already set it up. You can save your experiment as a template, just run that experiment, and then you can just wait for your acquisitions to be done over different days. What you need to do as a user regularly is every second day, you need to come in and change the media, which is usually what you do with your cells, just feeding them with fresh media every second day or every other day, depending on how your assay is being planned. And once you validate your tissue is mature enough, you can go into your endpoint assays. which includes pharmacology assay, but also for downstream assay imaging or molecular studies. So usually in a standard assay, which we do in our lab, it takes up roughly somewhere around 20-22 days before we reach into our endpoint assays. You can also have a shorter days, but this is our standard assay which we are running in our lab. So showing and data. So starting with a quick example of here you can see a quick overview of a 96 well plate, a full plate being seeded with the tissues. And we got an 89% success rate in seeding these tissues, which also excludes some of the tissues could be damaged for some reasons, or even some tissues not being formed. So excluding all those, we had roughly around 89% success rate. And the cells and the gels, initially, this is day 0. So you see just as a black blob in all those mini wells. In day 5 itself, you can see the tissues, the cell gel starts compacting and form tissues between those two pillars, which I showed you earlier. And what happens is, this is a cardiac tissue. So they start contracting these tissues. And what happens is there is a camera, and this camera starts acquiring this movement of these pillars. And since we know the stiffness of the pillars and we control the stiffness of these pillars, we can extrapolate what is the force generated by tissues over different days. So here is an example where we are following the tissues for different days. So here you can see on day 0, which is like cells everywhere. Day 1, you can see the edges are falling. And by day 2 itself, they already formed a piece of strip between these two pillars. And they start having spontaneous, we usually see spontaneous contractility by day 5. And then we start pacing those tissues for 2 weeks before we go into the endpoint assay. So here you can see on day 2 itself, there is very small spontaneous contractility beat, which improves significantly by day 5, even spontaneous, just spontaneous. And then it improves further with starting on the pacing within just 2 days. You can see how the pacing, the force has increased, and even the contractility profiles like the pulse width have improved just with 2 days of pacing here. So in our lab, we have continuously assays running, not just in the lab, but also at the platforms at some of our customer sites, where we have kept the tissues even as long as 60 days in this plate. And we just terminated it because we wanted to do an endpoint assays. So the tissues could be stayed. You just need to replenish it with fresh media every second day. So one of the key important of having these engineered tissues marker people would like to see is the maturation. Is my tissues mature enough to take into the next phase of endpoint assays or doing the pharmacology? So one way you functionally validate your tissues, whether it is mature, is force-frequency relationship. So basically, for those people who doesn't know quickly about force frequencies, basically you try to pace your tissues in different frequencies, increasing frequency, and see if your tissue is able to follow it. And if they are able to follow it, that is a sign that the tissues have, at least for the cardiac tissues, they have matured and they have much proper calcium dynamics in structures and all these, that it can sort of follow the tissues. But always there is a limit in the biology, right? So it do follow at a certain frequency, and after that, it starts dropping down. It shows a negative relationship. And this is exactly what we see in our engineered heart tissue. So here on the black on the top, you can see the pacing of a tissue from 0.8 hertz all the way to the 4 hertz. And you can see the amplitude increasing over time and then getting short. That's also shown in the normalized contraction below on the left side. The contraction peaks around 2.2 to 2.5 hertz. Actually, there is a published data which on human data, where in the clinics they have compared a force frequency relationship between the control and a heart failure patient. You can see even the control patient, it peaks around 2 to 2.5 hertz frequency. So this lines up, your engineered tissues are also more sort of matured up in a line with what you see in more mature systems like humanized systems, and sort of gives you the confidence to move your tissues into next phase, that is more the assay phase, which includes the pharmacology. So coming down to the pharmacology, we would like to see its response to your inotropic drug. So here is an example of isoprenaline, which is a positive inotropic drug. You expect to see a increase in the contractility amplitude, but also you would like to see a chronotropic effect, that is increase in the beat rate. And this is here, you can see the force increases in a concentration dependent manner, while that is the inotropic effect, while you also see a chronotropic effect that in a dose dependent way, you could also see an increase in the beat rate. So your tissues do show the responses to these two inotropic and chronotropic with isoprenaline in a dose dependent way. Coming down to the negative inotropic drug, which is shown as nifedipine, which is a calcium channel blocker. And you can see here also, you can see a decrease in the amplitude of the engineered heart tissues with the force in a concentration dependent way. And as I mentioned, our cameras are acquiring at a very high frame rate. That also helps you when you're working with higher doses. You could also pick up these small extra beats in our signals here. So that also shows the sensitivity of the system or the platform to pick up these extra beats in your contractility peaks. So these two are a quick example to show you how our platform shows the maturity, but also pharmacological responses to the standard drugs. I'm not showing here the other drugs, but we also have tried E4031, cisapride, and also some of the other sodium channel blockers on our platform. To sum it up, as we have seen, the whole workflow sort of covers up with the pipetting robot and the software taking care about the automation, helping you to having more reproducible data, but at the same time making sure you have that scalability, having that high throughput with the 96 well plate, but also managing multiple assays in multiple slots with multiple different pacing protocols simultaneously by different users gives you a huge big benefit, having that metrics in your platforms there. And having that standardized SOPs of cell gel casting and having that pipetting robot really helps you to get more mature tissues, having all those checking into those maturity markers functionally, and also having that responses to the pharmacological compounds. So that's going to help you to advance your drugs and your drug assays from different levels, not just for the cardiac safety assessment, but also to help you to develop your disease models, which nowadays is growing demand for personalized medicines. You need to have, with these iPSCs, you can have patient-specific cell types. So you can also use this platform, not just for screening purpose or the safety purpose, but also for designing or having disease modeling systems or assays in your workflow. So go ahead. Thank you. I would like to hand over the slide back to Damien, and would be really happy to take on any questions from here. Thank you very much.
Perfect, Lijo. Thank you. And you should come back to the stage because you're going to be the one answering the questions. I definitely don't know the answers to these questions.
Sure.
There you go. Thankfully, he's back on stage. Otherwise, I would have been a little bit short for the answers. And thanks for coming back on stage as well, Dr. Vaidyanathan. So we've got quite a number of questions coming in. So I don't really want to stick around too much longer with me rambling. Let's get straight into the questions. And again, you both should be able to sort of see this in your own chat boxes on the right. So if my accent is throwing you in any way, you might be able to read them for yourself. So I'll start off with what came in first earlier on, or actually this is in reverse order, but I'm going to go to the bottom. So Brian was asking earlier on, what is the estimate of cells per engineered tissue compatible with the chips? That's probably for you, Lijo.
Yeah, so we roughly now can go less than 50,000 cells. It also depends on, we have tried the engineered heart tissues, which is less than 50,000 cells per engineered tissues. you can use. And if it is skeletal muscle cells, is even less. It's somewhere close to 15 to 20,000 cells per tissue.
Great. And again, I'm going from the bottom up, so in chronological order, in case you're wondering where these questions are in your question box. Stephen did ask, what is the sampling rate of the camera? But this was answered by Sandra at 150 frames per second. So we've got that one covered. Ronald asks, how do you convert the optical measurement into the contraction-like traces shown? And again, that's probably more on the technical side for you, Lijo.
Yeah. So these are optical-based systems, and these we track actually the pillars, the contraction over the pillars or the tissues, and then that's how it is being translated into contraction pieces or the traces, which you look like. Yeah, and since you know the stiffness of the pillars, you can actually estimate what is the force generated by these tissues.
Okay, and then we've actually got two questions from Stephen, and they're interrelated, so I'll almost ask them as a two-part question. And this might be to both of you, but certainly Lijo, and potentially if you want to chip in, Dr. Vaidyanathan, please do so too. So Stephen's two-part question. After 2 weeks of pacing, if you stop the pacing to the cell, do they still show intrinsic beating, or are they quiescent, which is the first part? And then I think almost as a second part to that, once matured after 2 weeks of pacing, and you can see a positive force frequency response, if you then stop the pacing, how long does the maturation be maintained? For example, if you stop pacing for 1 to 2 days, is the positive force frequency response then lost?
Good. Thank you. That's an interesting question. Starting with the first one. So after 2 weeks, if the pacing is stopped, if you still see the spontaneous beating, so what we have seen in our experience, it's dependent very much on the cell source. So some of the cell type or the cell sources we got, some of them become silent after 2 weeks of pacing. They don't have any spontaneous beating. While some cells have the spontaneous beating, but actually a much lower frequency or a much lower rate, UDA spontaneous. So we have seen that that's very much dependent on what is your cell source. Because we have I have shown only the data from the FUJIFILM, but we have also worked with cells from different sources, from academic labs, but also the commercial ones also. So it very much depends on that. So going to the second one, if you stop the pacing and you see the force frequency relationship, so I would say I would not assume that the maturation to be reversed. So if it is mature, it should be mature, but we have not tested like stopping the pacing and then trying the force frequency to assess whether it's positive or not. We have not tested that particularly same. But I would assume that if the tissues are mature, should not be reverse mature. Correct me, Ravi, if you have some more inputs for that from yourselves.
Yes, so sure, thank you. So I can talk about what is published, right? And so the one paper that comes to my mind is the BioWire paper, where engineered heart tissues were developed using our cardiomyocytes. Their protocol is longer than 2 weeks. I believe they pace for about 4 from the publications that I've read. And I think they do see an increase in IK1, which is what I showed on my last slide, which actually makes the tissues quiescent. So at least there is evidence that our cells, in combination with fibroblasts on an engineered heart tissue platform, should stop beating spontaneously. And then there is pacing involved there too, but I think they ramp up the pacing over time based on that protocol that is published. So yes, so they do get mature, and this is very important for them to stop spontaneously beating, so that you can pace them at lower and higher frequencies. Right? And I think that is where this question is coming from, because for a force frequency response, you need to go over multiple frequencies. and I think our cells definitely have done that, but in a longer time frame from what I can say. And coming back to what Lee Jo also said, so we see that also on the cardiospheres. The cardiospheres where there is a combination of 3 cell types. We see that the beat rate is lower than in 2D, and it is around 20 beats per minute by 2 weeks when we recommend that you measure calcium transients or whatever you're doing with them. That is when you have, that's the spontaneous, but it is slower, definitely.
And just to add to that, Stephen has also got back almost with some information around maybe where some of those questions came from, because he's saying that they've seen it reverse within about 24 hours after the electrical pacing has been stopped. I don't know whether that's similar to maybe some of your studies, but certainly that's what Stephen has seen.
No, we have not particularly checked for.
Okay. But I can tell you in 2D, definitely that happens. I don't know if, Stephen, you're talking about 3D, but in 2D, definitely when you have monoculture, as soon as you stop pacing within 24 hours, they do reverse back to their original phenotype. In 3D, yeah, it's most people try to run their studies within 24 hours after doing a 2-week pacing study. So that comes back to Lee Jo not looking into that extensively, probably.
And Stephen does have a further question. So, have you profiled a large number of diverse positive inotropes in this system and compared the responses to ex vivo human tissue data, for example, from literature, to understand the translation?
For us, on our platform for now, we have tried mostly the standard compound, that is the isoprenaline. I have tried nifedipine and some of the other drugs. Particularly about the positive inotropes, that's something in our panel, which we are actually planning to run up soon. Not just our site, also few of our customers are also. And
while we're waiting for questions to come in, I probably have one for each of you too. So I will go with I'll go with my first question to Lee Jo. You talked about pacing protocols, and I'm completely ignorant in this, so sorry if this is a particularly stupid question, but you know who you're dealing with right now, so it might be a pretty simple, stupid question. It might be great for you actually, because you'd be able to answer it very quickly. So yeah, they seem like quite important aspects of what you can be doing in terms of maturation and potentially even presumably putting in a normal physiological pacing protocol versus a pathophysiological pacing protocol and anything in between. How important are they, and how complex can those pacing protocols become?
Super. No, that's actually a good question, because we have tried some pathological pacing frequency. For example, we worked with an academic group where we got atrial cells and we did atrial tachypacing also. So you can actually do such type of pathological pacing into that. And what we have done is pacings. What we see is pacing do help to improve some of the profiles. But we have also not said so. We also need to validate if pacing is required for maturation and how much. That needs to be validated. But we do always see pacing do help these tissues sort of having much more better responses to the drugs, and also these functional markers like force frequency relationship studies. If that answers you, Damien.
Yep, that's for my simple understanding. That's a great answer. Thank you. Philomene asks, how do intra-organoid diffusion barriers influence the effective drug concentration at ion channel function, and do these lead to misestimation of ion channel modulation? That's potentially for both, to be honest, but whoever wants to field that one.
Since it mentioned about organoid, I think so. Is it indicating to Ravi? I don't know. But
So I think I can take this one. I'll start at least. So I think the thickness of the tissue is what is being referred here, because the diffusion of compounds to the interior is important, because there is no vessels here, right? That takes them where they usually go in a physiological system. So for the cardiospheres, I can tell you the size of it depends on what you're doing. If you're doing it in a 96 well plate, we recommend that you use 10,000 total cells in a well. So that includes cardiomyocytes, fibroblasts, endothelial cells, and the thickness or the diameter of those tissue is about between 400 and 450 microns. And then if you're doing it in a 384 well plate, we recommend that you use 5,000 total cells, and they tend to be more in the 350 to 400 micron range. From what we are seeing is that the drugs definitely are on the outside. They have an effect, but because these are also contracting, I feel there's some movement inside, but it's very difficult to tell. But what we know is we don't have a necrotic core, which is what usually people are worried about when your tissues get thick. And if that can have a strain on your system affecting your contractility readout. But yes, we do not know if it makes all the way to the inside, but given that cardiomyocytes are 65% of the population, they're the majority, most of them, a lot of them are also on the outside. So you start seeing an effect right away. And that is what we see in our spheres.
I can add to that one from our platform, Ethica. As Ravi also mentioned, necrotic core is quite critical. And the benefit of having this tissue, or the main goal of having these small mini wells, is actually to keep the cell numbers low. So your tissues are not very thick, staying inside the oxygen diffusion barrier. And that helps us to actually have a much more drug exchange throughout the tissue. So we have some stainings of the live and the dead tissues, and we don't really see also necrotic core inside our tissues, engineered tissues also. So it should be having a better diffusion.
And I can just add to that as well that, yeah, effectively what you were just saying in terms of the small number of cells, or the small volume of cells effectively, Lijo, means that diffusion barrier of things like oxygen and other nutrients is really quite limited or minimal. So it's not such an issue that you don't have, for instance, a blood vessel feeding a thicker diameter. And Sandra, one of our, the director of this whole project, so certainly the person who would know, she's suggesting that diameters of the tissues are between 300 to 400 microns. So those diffusion barriers are relatively limited because of the relatively small volumes that you're dealing with these 3D tissues that are formed on the Ethica. And I can move on to some more questions. So Serge asks, do the long time pacing of iPSC organoids affect cardiomyocyte morphology?
Is it? I don't know. Ours are not organoids. Ours are cells compacting form the engineered tissues. Yes, one of our PhD student early has done this work where they have done this tissue staining, but they are not compared with and without the pacing. But they do see more alignment of the cells in these engineered tissues. We have not particularly looked in if the pacing facilitates that one.
Okay.
I can talk a little bit about.
Sure, Ravi, add to that.
Oh, do you want me to add? Sure. Mainly ours are not we don't call our spheroids organoids because they're mostly assembloids. Organoids inherently have a structure, right? At least by definition, but I know people use it very loosely nowadays. But we do see some alignment that in our spheres too, where we see sarcomeric alignment, and we also see that in 2D actually when we combine cardiomyocytes and fibroblast and long-term culture. So just wanted to add that quickly.
Great. And a further question from Ronald. If you do not pace, do you also see the positive inotropic and chronotropic effects of isoprenaline or other drugs that potentiate contractility, myosin activators, etc., for example?
I can take that one quick because I did have data where with the cardiospheres, we do not pace. I mean, you can pace them if you have the right technology, but it's not as easy as in the Sophion system, I can tell you that. You'll have a lot more wires hanging around. But when we don't pace it, in the triculture spheroids, we do see positive inotropic effect. That was one of the data slides that I showed, where we tested at least 3 different positive inotropes, and they all had a chronotropic and inotropic effect on the cells.
And so I am aware of the. Oh, sorry, Ravi. Please go on.
No.
Oh, not me. It was Lijo.
Oh, Lijo.
No, it's just to add to that one. In our normal workflow, we don't. We usually pace our tissues and check for the compounds effect there. Yeah.
And I am aware of the time. We obviously have now reached the hour mark. I could potentially ask another question of Ravi. Problem is, my questions are never particularly short, simple, and sweet. I never can formulate them into anything other than a mini essay. So with time potentially against us, maybe I should save my question for Ravi, for offline, because we have reached the hour mark. I'd like to thank both our speakers, Doctors Lijo Cherian and Dr. Ravi Vaidyanathan, and for both of you for giving your time and your expertise to this subject. It's very new to me, as you might realize, but it's been really interesting nonetheless. I hope the people that be out there in the world watching all of this have got it in place. And of course, if you've got further questions, don't hesitate to email us so that we can answer your questions, either if we haven't answered them in enough detail already, or we can answer new questions that arise after this webinar. And again, just to remind you, this webinar has been recorded, so you can get the recording as and when you want to. If you couldn't catch it all or you've missed some of it, you've had to take off because we are running a little bit over time now. But yes, that will be made available as a recording after the webinar. Thanks to the speakers again. Thanks to all my colleagues in the background, the unsung heroes that rarely get to sort of take the applause. They've all done a great job in making this webinar happen. I'm purely a spokesman for them, so they're the real heroes and the people that make this thing work. Thanks to everyone, and take care, and we'll hopefully see you at the next webinar in the future. Goodbye for now.
Goodbye.